Hi. Thank you for the invitation, everybody, and thank you for coming along. So what I'm going to talk about today is what I guess has been a bit of a side project for me. Most of the work going on in my group at the moment on Soay sheep is involving epigenetics and methylation and methylomes, but that's very much a work in progress. So today I'm going to talk about something that's been a bit of a side project, and that is a rare polymorphism in Soay sheep. And in particular, it's the presence of white patches on, in this case, on this lovely young ram's horns and on his face. And I'll explain in a minute about why this is a polymorphism that we haven't really worked on much in the past on this long-term study. But before I do that, I should explain a little bit about the project. I'm obviously in a place where everyone is fans of long-term studies and familiar with them. But the Soay sheep project is one of the most detailed studies of any wild mammal anywhere in the world. The study's been running since 1985. And all the Soay sheep on the island of Herta, which is in the St Kilda archipelago, which lies off of the coast of the Hebrides here. All of the Soay sheep in the Village Bay area of Herta have been tagged as lambs and followed throughout their lifetimes since 1985. I say all, nearly all. And this means that there are detailed records of life history and genetics and parasite measurements, etc., of around about 15,000 sheep that have been born in the study area since then. The reason why people first started studying the Soay sheep project was mostly because of its population dynamics. In the early years of the project, people thought the population went through periodic cycles where the population would grow for 3 years and crash after the 4th year. That was quite rapidly shown not to be the case. The population does still undergo big mortality events in some years. In general, the population has grown in numbers. The whole island population is typically around about 1,500 sheep. Now, Soay sheep are probably the most primitive breed of sheep anywhere in the world. They're thought to have been on this group of islands for probably 4,000 years. And the people that lived on the island until 1930 actually replaced Soay sheep with more modern breeds. So on Herta itself, they were farming more modern breeds, and Soays were only on the island of Soay until 1932, when about 100 of them were moved onto the main island of Herta, where our study site is. And this becomes a bit relevant towards the end of the talk, which is why I mention it now. But essentially all you need to know at this point is it's a long-term study similar to the ones that you'll be familiar with detailed life history data on lots of individuals. We started genomics work rather than sort of more molecular ecology and genetics work around about 2000 when SNP typing came along and whole genome sequencing was becoming feasible, wasn't really routine at that point. But one of the first things we did in the population, I should say the Soay sheep project is involved many, many different researchers and different PIs. It's led mostly from the University of Edinburgh these days. And a lot of this work is collaborative between ecologists and geneticists, parasitologists, etc. But those of us interested in the genetics, the first thing we really started to look at once genomics tools became possible were 3 very striking polymorphisms in the population. There's one for coat colour. So about 3/4 of the sheep have dark coats, which is the ancestral type. These look like mouflon, the ancestor of all sheep. But 1/4 of them have these light coats. There's a coat pattern polymorphism. Most sheep have what we call a wild type pattern, where you'll see the lighter belly and around the bum and around the face. But some of them have a uniform self colour all over. And perhaps best known of these is a horn polymorphism, which is under heterozygous advantage in males. Males have 2 morphs. They have large curling horns, or what we call skers. In females, there's 3 morphs: horns, skers, or the absence of horns, polled. So in the early noughties, up until I guess about 2010, we produced quite a lot of papers identifying initially the genes responsible for these phenotypes, and then studying selection at these genes. And these were, I guess, the low-hanging fruit for genetics work in this population. We subsequently moved on to the traits which perhaps are more typical of most traits that we study in the wild, traits which have a more complicated polygenic basis, and they're altogether more challenging things to look at, but arguably more rewarding when you do get some interesting results from them. So we sort of parked the single locus polymorphism stuff until very recently. And the polymorphism I'm going to talk about today is this presence of white patches either on the skin or sometimes on the horns, sometimes on the hooves. And this is barely a polymorphism in the sense that certainly when I was an undergraduate, I was always taught a polymorphism had to have a rare form that was present in more than 1% of individuals. And these white patches are found in about 1% of individuals in the population. So it never really seemed like something we could really do any meaningful studies on because there just weren't enough individuals in the population that express these phenotypes. This is quite an extreme one here. This is a male. This picture was taken during the breeding season in the early 90s. And it's unusual to see animals with as much white markings on as this. But over the years of the study, we began to see, a reasonable number of them. Here's fresh-faced Dan Nussey, holding one here with a white top knot. A white top knot's perhaps the most common of these genetic markings that we see. And here's a bit of a rogues gallery of white patched sheep. So quite recently, I sort of thought, I wonder how many of these sheep have now got these white markings, and dug into the database, and it turns out there was, a reasonable number. So I thought it'd be quite interesting as a bit of a side project to just explore the genetics of it a bit, see if there's anything interesting going on. And that's what I'm going to talk about today. So really there's three parts to the talk. The first part is exploring the genetic basis of these white patches, looking at the genetic architecture of them. The second part is to see if there's any evidence that there's been selection on the white patches in the population, and whether there's any evidence of molecular evolution of the genes that are probably responsible for them. And the last part is going back to the history of these white patches and whether there's any legacy of some of the other breeds that have been on St Kilda that are perhaps responsible for them. So I mentioned briefly earlier that sheep genetic tools became, or genomic tools in general, but sheep genetic tools became available relatively early on, I would say, compared to a lot of people studying wild populations. And that's because obviously there was quite a lot of investment in developing tools for domestic sheep. We're very fortunate that there's a SNP chip that has about 50,000 SNPs on it, of which about 36,000 are polymorphic in our population. And we still routinely use these chips to genotype every animal that's caught as a lamb. And one of the nice features of Soay sheep is that being a fairly isolated population and a fairly small effective population size, linkage disequilibrium extends a long way across the genome, so between markers. So 36,000 SNPs might not sound a lot compared to some of the genomic papers you'll read these days, but it's actually sufficient to tell you about all of the different blocks of linkage disequilibrium in the sheep genome. It's very powerful for this system. We also typed a smaller number of sheep on a higher density chip, which has about 500,000 SNPs on it, and using imputation, we've got known SNP genotypes for over 400,000 SNPs in over 7,000 sheep. It's probably more like 8,000 or 9,000 sheep now. But for the purposes of this data set, we have just over 7,000 sheep that are going to be analysed in this particular talk. So once I realised that there were maybe a sufficient number of sheep with these white patches to do some genetics on it, I decided to see if we could find any evidence of what the genetic basis for it might be. So I took a GWAS approach, but this is a fairly complicated trait to study because although it's probably caused by a small number of genes, it's most likely, it does seem to run in families, it's not what we call fully penetrant. So by that, what I mean is an individual could have the genotype that confers our phenotype of interest. You might expect it to be a mutant, but you don't always see that mutant phenotype. And that's probably because these white patches are certainly they're formed when the animal is still in utero. It's probably a very brief period where the melanin synthesis pathway is not functioning normally when it should be. It must be something that's regulating a gene involved in that pathway, presumably. And maybe that switch of whether it's switched on or off is determined largely by a small number of genes, but it may be that there's a very fine window in which those genes have to be switched on or off for the phenotype to emerge. So it's not what we call a fully penetrant genotype. But fortunately, we can still try and model that in a GWAS type approach. And in particular here, for those that are interested in these things, I use an approach known as a mixed linear model-based approach. This means we can control for relatedness of all the different animals in the population when we test each SNP one by one. We can use an approach known as leave one chromosome out, so we're controlling for the effects of any other chromosomes when we test each chromosome. We can treat it as a presence-absence trait, but we can set the prevalence to 1%, which is about what it is in the population. And these approaches deal quite well with phenotypes that are not likely to be fully penetrant. So that's just a bit of the details in there. Basically, it's a mixed model. It's a binary outcome, and it's controlling for the fact that our population are not a bunch of unrelated individuals, which is what you typically see in a genome-wide association study, but obviously many of them are closely related to one another because they all come from the same closed population. Anyway, did this GWAS, and amazingly, there are some really, really strong peaks. So this dashed line here is something you might expect once by chance in the genome. The bold line, only 5% likely to happen in a whole genome scan. So we've got these two enormous peaks here, plus some other regions which look quite interesting as well. But two very, very strong peaks here. The p-value is 1 × 10⁻⁻60 of this higher peak. So despite there only being 100 or so individuals with this phenotype, there's still sufficient power to find SNPs which are very, very strongly associated with it, which is rather gratifying, of course. And if you zoom in on it a bit more, both of these peaks are on chromosome 19. And you can see that they're quite a long way apart on chromosome 19. They're about 30 million base pairs apart. But they are two very strong peaks. And they appear to be independent of each other. I won't go into the details of why I'm pretty convinced they're independent of each other. One suggestion could have been that maybe the Soay sheep genome is different to domestic sheep, and the assembly of domestic sheep isn't quite correct for Soay sheep, and maybe this bit here on Soay sheep is actually next to this bit in Soay sheep, but not in domestic sheep. But I probably won't have time to explain why I don't think that is the case. I think these genuinely are two different independent peaks. The peak on the left, the slightly lower of the two, there's not really any good candidate genes in that region. There is one gene here, but there's no evidence that this is associated with any part of the melanin synthesis pathway, and the nearest peaks to it are not right under that gene anyway. Along here, we have a much better candidate, the melanocyte induction transcription factor, or MITF. It's a really big gene. It's about 200,000 bases long, this gene. But you can see that the main peaks are right along it. The gene is read from this direction to that direction. So these are all SNPs around the later exons in that gene. And this gene, melanocyte induction transcription factor, some of the associated phenotypes shown to be with this gene in other systems, shown to be associated with this gene in other systems, are remarkably similar to the phenotypes I've been describing. So this is a phenotype in horses known as splashed white, and you can see it's very similar. Here we've got some mouse mutants. We've got similar phenotype here, and this is a human with something called Waardenburg syndrome. Von Willebrand syndrome. I can't remember which of them now, actually. But the reason this appealed to me, that this was one of the phenotypes that was associated with it, was back when I was an undergraduate, a long time ago, in the late 80s, early 90s. I remember the first human genetics lecture I ever went to, which was quite a formative experience for me, because I was a biochemistry student at the time, and I switched degree as a result of it. But the very first Mendelian trait I was taught was this particular disorder, and at the time the gene responsible for it wasn't known, so it feels like it's come full circle. So MITF seems like a very, very good candidate. More recently, a study's come out just this year or the end of last year, showing that the same gene, not the same SNP, the SNP's about 20,000 bases away, is associated with variation in hoof colour in Australian white sheep. And actually, hoof colour is one of the hooves are one of the areas in Soay sheep where you sometimes see a white hoof or sometimes just a white toe in the animals that have these white mutants. So it all fits very nicely that this is our causal gene. I'm actually not too bothered about finding the causal gene or the causal mutation, but it all makes it more convincing, I guess. The gene itself, melanocyte-inducing transcription factor, plays a key role in the melanin synthesis pathway. It actually interacts with one of the genes that explains both of the genes that explain our other coat polymorphisms, including TYRP1, which explains the dark light coat polymorphism. So it's all part of that same melanin synthesis pathway. I think it's worth actually just tabulating the effects of the top SNPs and giving you a sense of how much they explain the phenotype. So what I've got here is this is a table for the top SNP under each of those different peaks. So for the first, and basically what I've tabulated is whether that animal lacks white patches, or whether it has white patches, and the 3 different genotypes for the SNP. So what you can see here is that if an animal is AA, which is the more common allele at this SNP under the main peak, there's only less than 1% chance of it having white markings, 1 in 147. If it's a heterozygote, it goes up to 1 in 38, and if it's the other homozygote, it's about 1 in 4. So remember I said this isn't fully penetrant. So if you're GG, you're not guaranteed to have white patches. In fact, you're more likely to not have white patches, but you're a lot more likely to have white patches than for example, an AA animal. And similarly with that other SNP, the one at the start of the chromosome, the genotype associated with the white patches is this GG one. You've got about a 1 in 13 chance of having white patches if you've got GG, and only a 1 in 182 chance if you're AA. So you can have the underlying genotype without having white patches, and most of the time, if you have got that genotype, you still won't have white patches, which is why I think it must be something that is this gene must be switched off in some cells very transiently during development, if at all. I was interested in whether the 2 genes might interact to enhance the frequency of white patches, and there is some evidence of that. So if you take animals which are GG at the first SNP and just have the presence of at least 1 G allele at the second SNP, then the probability of having white patches goes up to over half. So those 2 SNPs do seem to interact. They're largely independent, but if you fit a model with an interaction between them, it is significant, and it slightly improves the model. So there's possibly some epistasis between those 2 different SNPs. Like I say, I don't think they are, actually much closer to each other in the genome than the sheet genome assembly would tell us. I don't think it's something as simple as that. And it seems a bit of a stretch to imagine that the first SNP, which is 30 million base pairs away from the other one, is somehow regulating it directly. I don't think it's a promoter of that gene or anything like that. Although I must admit I'm not enough of a molecular biologist to know if promoters can be 30 million bases away, but maybe that is possible. Okay, so to summarize that first part, there's 2 regions on chromosome 19 that play a major role in causing these white coat patches. They don't completely explain it, but they certainly, make a massive difference as to whether or not an individual is going to have these white patches. And one of them is a very strong candidate gene. The other one doesn't have any good candidate genes in and around it. And I think they're largely independent with some epistasis between them. So for the second part, I want to consider whether there's any evidence of selection, either on the phenotype itself or on those SNPs which are partially responsible for the phenotype. And I've looked at this in 3 different ways. First way is to just consider the individual fitness of each individual, so it's lifetime reproductive success. The next way is to look at whether the frequency of those causal alleles has changed over the lifetime of the study. So this is over 40 or 50 years now the study's been running. This data set's more like a 40-year window. And the other approach is going back a bit further in time and asking whether there's any evidence of selective sweeps in and around those causal loci. And I'll briefly go through all 3 of them. So lifetime reproductive success, we're just defining here as the number of offspring an individual has over its lifetime. We're not including whether that individual survived to year 1 or whatever. But they're pretty horrible distributions to try and model. In both cases, there's a big excess of zeros, but there's also over-dispersion as well, particularly in males. The most successful rams in the population have had over 100 offspring in their lifetimes. Males and females are precocious by the time they reach their first winter, so they can both reproduce as lambs. Or in the case of females, have their first lamb when they're a year old. So I've modelled both male and female lifetime reproductive success, but in different analyses for each, because their distributions are quite different from each other, even though they're both over-dispersed and zero-inflated. I've used the Bayesian package BRMS, which is very handy for this kind of approach. So it's been modelled as what's known as a hurdle negative binomial model. So the hurdle bit refers to the fact that they've got to essentially get over the hurdle of whether they've bred or not. The ones that haven't, that explains the excess of zeros. And otherwise, it follows more or less a negative binomial distribution. I've included in the model birth year as a random effect, and also whether or not an individual is a twin. Because if an individual, twinning is fairly common in Soay sheep, much less common than in domestic sheep, but maybe about 15–20% of ewes will produce twins each year. And if you're born a twin, that gives you a disadvantage in later life. You've obviously got some competition, which is going to mean you're going to be disadvantaged when it comes to reproduction later in life and survival. So if we look at females first of all, there's basically three plots here. The first one is just comparing presence of white patches with absence of white patches. We've got the mean lifetime reproductive success as estimated by the models on the y-axis. So in the case of females with white patches do have marginally higher, greater lifetime reproductive success. The 95% credible intervals for that are just above zero, but only just. And bear in mind, this isn't going to be, there's obviously the big error bar on that. So there's possibly some evidence that these white patch females are doing better, but I'm not particularly convinced by that. And then we've got the two SNPs. I think I've actually labelled SNP1 and SNP2 the other way around as to how I was talking before. This is the one at the start of the chromosome where I'm comparing AA just against the presence of a G. Absolutely no difference there. And if you take the other SNP, the one that's in the MITF gene, no difference between the three possible genotypes there. So none of the contrast between the SNP genotypes is significant. There's possibly some evidence that the patch itself has a fitness advantage, at least in females. In males, it looks similar in terms of white patches. That contrast is not significant. Those 95% credible intervals overlap. There's no effect on either of the SNPs. So, I would say on balance, certainly if you look at the underlying genotypes, there's no evidence of selection on the underlying genotypes under these peaks. There's possibly some evidence, if you treat it at the level of phenotype, that the animals with patches have got higher fitness, but it's pretty marginal. The next approach is to use something known as gene dropping, and this is using an R package developed by Susan Johnston in Edinburgh, who's the person that did all the very nice work on Soay sheep horns and horns polymorphisms some years ago. Susan developed this nice package which does what we call gene dropping. And what I mean by gene dropping is because we know the pedigree for this population, we can do two things. We can first of all, we can measure the change in allele frequency of those causal SNPs over time. And if the alleles associated with white patches have increased in frequency, then you might say they've been under positive selection. If they decrease, maybe they're under some kind of purifying selection. And what we can do is, because we got the pedigree, we can basically feed simulated SNPs down through that pedigree and look at the amount of change you get just by genetic drift. And it's using the pedigree to simulate that genetic drift because it's taking into account all the nuances of variance in male and female reproductive success. So the black lines represent 1,000 simulated gene drops of genes with the same starting allele frequencies, and the red line represents the actual observed change in allele frequency. This is for our SNP at the start of the chromosome. This is for the one at MITF. And essentially what you can see here is that the red lines are no different to what you expect from genetic drift. In fact, if you look at histograms of those regression slopes, where if you do a regression slope of allele frequency against time, you can see that the red lines fit pretty much bang in the middle of those distributions or close to the middle. So there's no evidence that these SNPs are doing anything that's not what you would expect from genetic drift. They're not increasing in frequency because they're favoured. They're not decreasing in frequency because they're selected against. And then the last approach is trying to study molecular evolution around these genes to see whether there's any evidence that there's longer term selection acting on them. And there's a little thing we have to do first to make that possible. We have to first of all phase the data. And this is also relevant for the last part of the talk where I talk about the history of the polymorphism. And by phasing the data, I'll explain briefly what I mean by that. So let's imagine we've got an animal here and we know that at SNP1 it's got alleles GA, and allele 2 it's got SNP2 it's got alleles C and T. There's two possible ways in which it could have those genotypes. It could have G and C on one chromosome and A and T on the other, or it could have G and T on one chromosome and A and C on the other. So these are the haplotypes that an individual has. And a lot of the molecular evolution analyses that we want to do, we need to know haplotypes. So we need to basically know which alleles have been co-inherited on which chromosome. And this program ShapeIt is very handy for doing that because we've got quite dense marker data. We can use all that marker data. You can use the pedigree information as well, which helps. So essentially the first step is to take our, in this case, just over 6,000 animals that would phase genotype to have known coat type and try and work out the two haplotypes per individual. So in other words, work out whether they've got this configuration or that configuration and work out where recombination events have happened during the transmission of alleles from parent to offspring. So it's a slightly complicated phase, but it's essential to do the next part. So taking that phase data, we can then use various tests that look for signatures of selection. And in particular, the one we've used here is something called the integrated haplotype sharing score. And I'm not going to go into the mechanical details behind that. Some of you might be familiar with the work that Pardis Sabetti has done looking for signatures of selection in between human populations, and something quite a well-known test known as the cross-population extended haplotype homozygosity test. This is an analog of that you can apply within a population rather than when you're comparing two populations. And essentially, you measure it in sliding windows. You walk along the chromosome. Here I'm just showing chromosome 19. And you can convert that into a Z-score and a p-value. And anything outside of those dashed lines is evidence of a positive selective sweep. And anything within those dashed lines is there's no evidence of selection going on. So our SNPs of interest are about here. And unfortunately, the other one is at about 2 million bases at the start of the genome. And it's quite hard to phase our data set at the very start and end of the chromosome. So we can't do this analysis for the SNP and the region at one end of the chromosome, but the one around MITF, we can do this analysis. It takes a long time to compute some of these, so I just took 500 animals at random whose allele frequencies were representative of the overall population, but it's still a decent sample size for this kind of analysis. And if you zoom in on the region of chromosome 19, where MITF is around 31.6 million base pairs. There's no evidence of selection going on here. Anything above that line would be evidence at not particularly strong threshold, 0.05, but it's not even at that. So there's no evidence that there's a selective sweep that's gone on in and around this region where MITF is. So to summarize that part, there's no real evidence of selection on the SNPs underlying this white patch phenotype. Possibly the white patch phenotype itself, there's weak evidence that females with the white patches, or maybe both sexes with the white patches, are slightly fitter, but I'm not convinced by that. The temporal changes of alleles are completely consistent with drift, but there's no evidence of a selective sweep at the region either. So I would say that this polymorphism is something that is probably neutral, and that's probably a combination of it being rare and the underlying genes not being fully penetrant. So even if you have got those alleles, you're not necessarily going to express the phenotype. And then for the last part, I just want to briefly talk about where the white patches might come from, a bit about their origins. And Soay sheep on the St Kilda group have a rather complex history, which I'll try and explain as clearly as I can. But it is somewhat confusing. I'm going to walk backwards in time rather than, I think it's easier to explain it this way. So St Kilda is actually a group of an archipelago of 4 islands. The largest island where our study site is this one, Herta. Soay itself is this one. And then there's an island a few miles away called Borroway, which has a different breed of sheep on it. And I've used different colour schemes to represent what sheep are on the islands at different times. So in the present day, we've got Soay sheep on Herta and on Soay, and we've got Borroway sheep on Borroway. But if you go back to 1932, there were no, so the permanent human population of Herta left in 1930, and they took their livestock with them. And 2 years after that, 107 Soay sheep were moved from Soay onto Herta, which is when our current study population was established. When the St Kildans were there, they didn't keep Soay sheep, they didn't keep Borroway sheep, they kept Scottish blackface sheep, which by this point was the predominant breed throughout the Highlands and Islands of Scotland. So St Kildans, when they were living there, were farming blackface sheep with them, and they took them all with them when they left. Before Scottish blackface sheep were introduced, they kept a breed that's now extinct called the Dunface, or the Old Scottish Shortwool, shown by the solid green colour here. So gradually St Kildans introduced Scottish blackface and replaced their Dunface with the Scottish blackface. But there is a written anecdotal story saying that they moved some of their Dunface, the now extinct breed, onto Soay in about 1850. They used to get taxed on the number of sheep they had, so they hid some of their sheep on Soay, where they would have had the opportunity to hybridize with Soay sheep. And before that, then there would have been, Soay sheep would have been completely isolated. So we think this introgression event happened on Soay, where Dunface, which are now extinct, introduced genes to Soay sheep. Dunface are now extinct. However, Borroway sheep were made by crossing Dunface and Scottish blackface sheep. So we have this population of Borroway sheep that will contain some genes from contemporary blackface sheep, but also some genes from this now ghost population. And what we've shown in the past is that Soay sheep on Herta have probably got some Dunface sheep genes in them that came about through this event here. And this is actually responsible for the dark light coat polymorphism, not the horns polymorphism. We think that's older than that. That seems to be ancestral. And it's also probably responsible for the wild type self-type coat pattern. So the two coat phenotypes we've looked at before, there's been an introgression event that explains the polymorphism, but for the horns phenotype, it doesn't seem to explain it. So I was interested to know whether that was the case for the white patches as well. So these are Borroway sheep. They're another rare breed, and they are a mixture of Scottish blackface and this now extinct Dunface Old Scottish Shortwool. The closest relatives to the Old Scottish Shortwool are probably the sheep breeds like Ronaldsay sheep found on some of the islands around Orkney. Oops, I've gone the wrong way. Okay, so what we wanted to do is see if we could test for any evidence of introgression at the loci associated with these white patches. So this is where we use this haplotype data. And the first step is to define what we call core haplotypes. So by that, what I mean is we take our SNP that's at the peak of our GWAS analysis, we look at the two alleles at that SNP, and then you look at the adjacent SNPs and you work out how many haplotypes the next 5, 6, 10 SNPs, whatever, explain, and you basically categorize those haplotypes into a discrete number of maybe 8 to 10, what we call core haplotypes. And then we look for those same haplotypes in all the other sheep breeds that have been typed. Because the same SNP chip is used across all sheep breeds, we can basically compare data across sheep breeds very easily. A bit like having genome sequenced for all the different sheep breeds. So we can look for those same core haplotypes in other breeds. And then what you do is you walk out from those haplotypes further and further, and you measure the distance to which they stay identical before recombination has broken them down. And the idea is that this extended haplotype sharing is an indicator of time since those haplotypes were shared between Soay sheep and whatever other breed you're comparing it to. And the hypothesis would be that if the haplotypes associated with white patches have extensive sharing between Soays and Bororé sheep, and in particular if the white patch haplotypes have got more sharing than the non-white patch haplotypes, then that would be evidence that this dun-faced breed had introduced the white patches into the Soay sheep population. This plot's a bit complicated. Essentially, each panel here is a different sheep breed that has been compared to Soays, and each bar chart represents a different one of those core haplotypes, and the y-axis is the length of this haplotype sharing. So the higher the bars are, the more haplotype sharing has gone on between Soays and other breeds, suggesting more recent ancestry in and around that region. This is Bororé sheep here. You can see Bororé sheep have got some bars which are higher than the other breeds along here, and the different colours represent the different core haplotypes. So we've got core haplotype 1 through to 8. And the asterisk ones are the ones associated with the white patches. The others are associated with not having white patches. And that was for the first SNP. I think this is the MITF SNP, and this is the one at the start of the chromosome. So in both cases, you can see Bororés have got some higher peaks here. But actually, if we zoom in on them, this first one is comparing Soay sheep to Bororés and Scottish blackface in the MITF region. What you'll actually see if you really dig into it is that the haplotypes associated with white patches in Soay sheep, they are these two light grey colours here. They'd be at positions 1 and 2 on this plot. They're completely absent. So Bororés don't contain those haplotypes, nor do Scottish blackface. So this would suggest that the white coat haplotypes have not been introduced from these ancestors of Bororés. There's no evidence of haplotype sharing, and it's the same for the other region as well. Is haplotype 4 associated with the white patches in this other region? Haplotype 4 is actually this one here. It is present in Bororés and Scottish blackface, but the extent of haplotype sharing is pretty weak compared to some of the non-white patch ones, and it's no different in Bororés than it is to any of the other breeds. So essentially what this analysis is showing is that these white coat patches are not a consequence of that event in the 1850s or mid-1800s when the St Kildans moved their dun-faced sheep onto Soay. It's not a recent integration event. So that means that these haplotypes have either been derived in Soay sheep at some point in their past and have been around for a long time, or they completely predate sheep isolation on St Kilda, and that they pop up now and then in breeds all around the world, and they're present in the ancestors of most modern sheep breeds. We can't really distinguish those two. But it certainly doesn't look like the case that this is a recent polymorphism that's arrived in the population through an integration event. It's been floating around for a long time, I suspect. Okay, so I'm going to wrap up there. So these white coat patches, they're barely a polymorphism. Their frequency is around about 1%. We think that at least 2 regions of the genome explain much of its inheritance, but not all of it, and I suspect that's because the underlying alleles are not fully penetrant. I think the polymorphism is probably selectively neutral. I should say, actually, in humans where this gene causes diseases, such as the one I mentioned before, there are other phenotypes associated with it, and in particular, deafness is quite common. I have no knowledge, one way or the other, of whether Soay sheep with white coat patches are deaf. I'm almost tempted to try and do an experiment where you can make a loud noise while filming a flock and see if the ones with the white patches are the slowest to react. It would be quite tempting to do it. Even if they were deaf, I'm not sure it would be much of a selective disadvantage on St Kilda. The only time I can imagine it might make a difference is lambs might struggle to find their mum if they get separated, but I actually don't imagine deafness would even be particularly strongly selected against in Soay sheep. I don't know. But I think it's, I think it is an example of a polymorphism that's persisted in a population that is selectively neutral. Normally when we study polymorphisms, it's because we're trying to understand what it is that maintains them in populations, and usually there's some kind of balancing selection or something like that going on. And here I can't see any evidence of any of the usual explanations of maintenance of a polymorphism. So I think it's probably just a combination of its rarity and its lack of full penetrance, and otherwise it seems to be selectively neutral. But I'd be very interested to hear other people's opinions on that. And yeah, I think thinking about it, it's quite rare to read papers where people actually talk about the neutrality of a polymorphism, because normally they try and seek some explanation for why it persists. So yeah, there it is. I should finish with some acknowledgements. In particular, Josephine Pemberton, who for many years has led the Soay sheep project, and obviously a lot of the genetics of it. We've collaborated a lot on that over the years. Dan Nussey as well, who's taken over a lot of the leadership of that project. The gene dropping work was developed by Susan Johnston. She's written this very nice package for doing that gene dropping approach. Xavier Ball is the main field assistant on St Kilda and is extraordinary. And Martin Stoffel is the person who did all that imputation, so increasing our 30,000 SNPs up to almost 500,000 SNPs. And of course, over the years of the sheep project, there's been many different funders. And of course, we'll acknowledge those as well. And thank you all for coming along. Very happy to take any questions.